Okay, so as Jim said earlier today, this session is going to be a little more molecular based and a little more basic science. Just to reiterate what Jim said, one of the big growth curves we had over the last 20 years is genomes and sequencing, and this is a slide that shows where we started. And as you can see, in September 95, we had the first genome, bacterial genome sequenced. It was Haemophilus influenza. But if you look at this slide, since then, the 20 years, we've now had over 14,000 publications on whole genome sequencing. So clearly the rise in technology over 20 years has been remarkable, even though, as we said, there's still a lot of challenges. One of the genomes that was sequenced, in fact, it was the second genome sequence, is RV. And everything we're talking about in this session is really based on our knowledge from RV sequencing, because it actually not only taught us a little about the basic biology, it actually uncovered a number of surprises. The biggest surprise was TB is a remarkably conserved genome. And because of that, it allows us, when we look at different nucleotide changes, everything, especially a non-synonymous change in a drug resistance gene, actually has meaning. And this is one of the unique properties of TB that we should take advantage of, and we have. So as an example, we know almost completely, except for some of the new drugs, we know the targets. So the real challenge was, if you could actually identify mutations in these genes, you could predict drug resistance and predict phenotypic drug resistance. So the real challenge was, could you develop a molecular test that could actually be discriminating at the allelic level? So as you can see in these few slides, you've heard this is the RPOB example, and as you can see, there's basically no difference when you're looking at the sequence of a wild type versus a mutant, except for a single nucleotide change. And if you have a tool that can detect it, you can predict rifampin resistance. The same is true for fluoroquinolones. Here's an example of a resistance gene, and it's a simple A to G mutation. This is what makes TB genotyping wonderful, and it's actually unique to TB, enabled to predict resistance. And this has been the paradigm that we've used for a number of these drugs, looking at different targets. However, like anything, and this is just an example of two companies who you've heard already, but this is the challenge for today's talk. There are paradigm shifts. The paradigm shifts has happened when we started to look at other targets. And the classical one that everyone probably knows about is pyrazinamide resistance, because pink A just does not follow the rules. As Jamie will tell you in a second, there are literally hundreds of mutations in the pink A gene, and we don't know what correlates with resistance. And therefore, the paradigm of using the RPO and gyrase idea doesn't work. And it also doesn't work for nitromidazoles, because we have about 5 or 6 different targets. So this is the challenge going forward, and this is the basis of today's session. So with that, I want to introduce the first speaker, Jamie Posey for the Centers of Disease Control, talking about pyrazinamide and defining the pink A mutations.